Welcome to Nikolai's Genetics Lessons, and in this video I'm going to talk about how to troubleshoot gel electrophoresis results. And today's question I got from one of my subscribers. I'm working on DNA barcoding for fish species using CO1 gene. In previous experiments, I successfully amplified CO1 gene using the same primers and PCR conditions, annealing temperature 52 degrees Celsius. However, in my recent experiment, after performing PCR and running the samples on the agarose gel, I did not observe any clear PCR bands. Instead, I noticed a sharp, unexpected band below the wells. Additionally, my positive controls also failed. So as you see, wells are here, and the sharp and bright band just below where DNA was loaded. And we do not see any sharp bands here at all, only something that looks like degraded DNA or contamination here. Those, as you see, there is bands present in a ladder. Looking at these gels, it is quite possible that the issue is related to primer degradation or template DNA quality. Even though you used the same primers and PCR conditions that previously worked well, primers can degrade over time, especially if they have been subject to multiple freeze-thaw cycles or improper storage. Degraded primers can lead to nonspecific binding and the formation of primer dimers, as you see on this picture, which often appear as sharp bands just below the wells, exactly as you observed in your gels. Here's another example of the primer dimers on the gel, which you can see as the faint bands. Additionally, the presence of smearing or irregular bands in some lanes suggests possible degradation of DNA template. If extracted DNA was exposed to repeated freeze-thaw cycles, or suboptimal storage, or contamination, it may have broken down into smaller fragments, which result in non-specific amplification and appearance of the streaks or shadow bands in the gel. And another potential factor is degradation of PCR reagents, such as Taq polymerase or DNTPs, which can also affect amplification efficiency, especially if these reagents have been stored for extended periods or suboptimal conditions. Since your positive controls also failed, this points more strongly towards a systemic issue with primers, template DNA, or reagents, rather than sample-specific problem. And finally, I would also recommend setting up a non-template control to rule out contamination or carryover from previous experiments, which can result in unexpected bands or artifacts. And also, you can get such results where your DNA doesn't move from the wells when you have protein contamination. So protein attached to the DNA doesn't allow it to move through the gel, and you basically would see a picture similar to this. And this is all for today. Subscribe and see you in the next video. Goodbye.